It is known how Staphylococcus provides itself with conditions for development in the epidermis. Staphylococcus bacteria manipulate the skin's natural protective mechanisms to provide themselves with optimal conditions for development. Scientists from the Intercollegiate Faculty of Biotechnology of the University of Gdansk and the Medical University of Gdansk have determined the study on the mechanisms crucial for the proper functioning of the protective barriers of the human epidermis was carried out in cooperation with the University of Oxford, the Karolinska Institute and Imperial College London. The results were published in the Journal of Extracellular Vesicles. The skin is the largest organ of the human body. An adult human has about 2 square meters of it. The epidermis covering it, made of specialized cells, keratinocytes, can have up to several layers. The most functions of which are performed by the upper layers of living cells and the upper stratum corneum, consisting of constantly exfoliating, dead keratinocytes. It is this layer of the epidermis that prevents excessive evaporation of water and is responsible for maintaining proper acidity and limiting the growth of bacteria, explains the Foundation for Polish Science, FNP, on the website. Filaggrin plays a key role in the process of building the stratum corneum. It is a protein that performs a number of functions in the skin, including is responsible for mechanical strengthening of the epidermis. It programmatically kills the superficial cells, forming the stratum corneum. This is a kind of life and death switch for keratinocytes. However, this process cannot happen too quickly. If it is activated too early, the barrier layer performing the necessary functions will not be formed. So the body must control the levels of filaggrin when it is produced in cells, explains Dr. Danuta Gotowska-Arziak, professor at the University of Gdansk and the main author of the study, quoted in the release. There is so much filaggrin in the upper layers of the epidermis that it forms granules that prevent intracellular release of the protein. Filaggrin is released from them into the cytoplasm only when the body is preparing to form the stratum corneum. Until now, however, it was not clear how cells from the deeper layers of the epidermis protect themselves from premature death caused by too high a protein concentration. And only the latest work of the Gdansk team showed that the deeper layers of the skin remove excess filaggrin using the so-called small secretory extracellular vesicles, SEVs. They are like soap bubbles sent by cells. It was once thought that they were simply used to remove toxic byproducts of the cell's metabolism. Today, however, we know that they also have another function. They can transmit signals between cells and tissues of the body. By reaching the circulatory system, they can even go from the skin to the brain, explains Dr. Gutowska-Owsiak. Researchers have shown that Staphylococcus aureus, a common bacterium that exacerbates skin inflammation, is able to manipulate this process. Filaggrin plays an important role in the prevention of atopic dermatitis. The less of it in the skin, the higher the risk of this disease. This protein controls the multiplication of Staphylococcus, because the products of its breakdown lower the pH of the skin, creating unfavorable conditions for it. In the case of a complete lack of filaggrin in the epidermis, the risk of AD is 150 times higher than with a normal concentration of this protein. In the latest study, the team of Dr. Gutowska-Owsiak showed that the presence of bacteria makes skin cells transfer filaggrin to SEVs faster. This causes the protein to be removed more intensively, as a result of which its concentration in the epidermis decreases. We believe that in this way, the bacteria can eliminate this protein from the epidermis, which impairs the functioning of the skin barrier. In this way, Staphylococcus manipulates the natural process to create living conditions for itself, describes the researcher, quoted by FNP. The findings could lead to new treatments for AD by manipulating the mechanism that regulates filaggrin levels in the future. They can also help in the development of new ways to prevent the development of allergies, which are also associated with pathological conditions of the epidermis. Dr. Hab. Danuta Gutowska-Owsiak is one of the winners of the FNP First Team program. The study was financed by the First Team program, as well as thanks to grants from the British Skin Foundation, Medical Research Council UK and the National Science Centre. Microplastic found in water trapped on plant leaves. Although we have been dealing with microplastics for a relatively short time, These plastic particles can already be found in almost every ecosystem on our planet. They have been discovered in the soil, in rivers, in our food and in bottled water. Recently, a team of researchers found microplastics in water trapped on plant leaves for the first time. Katerina Fogasova, Peter Manko and Joseph Abona from the University of Prešov in Slovakia initially set out to the eastern part of the country to study the organisms that live in the small pools of water that form on the leaves of aerial flowers. To their surprise, the researchers found differently colored fragments and fibers, some reaching 2.4 mm in length, which were identified as microplastics. The Dipsacus genus has characteristic opposite leaves that grow on the stem above each other in several tiers. Clamping on the stem, they form cup-shaped structures that collect water called phytotelms. This is a relatively common but overlooked aquatic microcosm with a very short lifetime of only 3 to 4 months, the researchers write. An article describing the discovery was published in the journal BioRisk. These phytotelms are very small and only occur for a short time. So the question is, how have they been contaminated with microplastics? The researchers ask. No other sources of contamination were found in the study area. So fragments and fibers most likely originated from the atmosphere. Another theory is that microplastics could have been transported by snails. The discovery of microplastics in small, short-lived bodies of water created by plants is further evidence that this type of pollution spreads through various routes and probably no environment on Earth is safe. Which of course makes our discovery quite depressing, the researchers write. The researchers believe that the discovery could be used to study when and how microplastics spread in the environment around the world. All because of the specificity of the phytotelm. They suggest that due to their abundance and theoretical ability to capture microplastics from the environment, phytotelms could be a good indicator of the presence of pollutants. Our publication, therefore, brings not only the first discovery of microplastic contamination of habitats of this type, but also the first proposal for a new approach to the use of phytotelms and similar microecosystems provided by plants, or artificially created, as bioindicators of the presence of microplastics in the environment, possible sources and routes of their spread and spatio-temporal changes in microplastic pollution, write the researchers. can tardigrades survive without water? Researchers have discovered the secret to their success. Water is a key ingredient for life on Earth. However, there are creatures that can do without it even for decades. We are talking about tardigrades. Scientists have finally discovered a trick these microscopic animals use to survive years of extreme dehydration. Tardigrades have been delighting with their ability to survive for years. They cope with various, extremely difficult conditions, often fatal to most other forms of life. It is conceivable that by learning their secrets, we could apply this knowledge to ourselves to make people more resistant to extreme temperatures, pressure, and even dehydration. For now, it's just science fiction. But scientists are still trying to understand the mechanisms responsible for their resilience, as it may have other benefits as well. These tiny creatures, up to a millimeter in length, live almost everywhere, but they prefer aquatic environments. For this reason, they are sometimes called water bears. They can withstand pressure greater than that found in the deepest part of the ocean. They tolerate high levels of ionizing radiation, exposure to all sorts of chemicals, and are the only life form we know of to have survived in space. They can also go without water for decades. Scientists have just described a mechanism that explains how these creatures can withstand extreme dehydration. While water is an essential component of all life as we know it, some tardigrades can potentially survive for decades without water. The trick is how their cells deal with this stress during the dehydration process, explains Takakazu Kunieda, a biologist at the University of Tokyo. New research on the amazing properties of tardigrades has been published in PLOS Biology. When these aquatic animals are in a dry environment, they shrink and take on a round shape. In this form, they become extremely stable and can withstand many extremes, including exposure to the vacuum of space. Scientists believe that when the water leaves the cell, some type of protein must help it maintain its physical strength so it doesn't collapse in on itself, explains Kunieda. So the researchers tested a group of tardigrades known for their ability to survive without water. In total, they identified 336 proteins that could be responsible for this phenomenon. After testing several different types, the researchers found that CAHS, cytoplasmic abundant heat-soluble proteins, unique to tardigrades, are responsible for protecting their cells from dehydration. Using experiments on human and insect cells, the researchers were able to show that CAHS proteins increase cell stiffness, helping them to resist shrinking due to water loss. These proteins also protected the cells from excess water. Attempting to check how CAHS proteins would behave in cultured insect and human cells posed a challenge for researchers. Because the method of staining them involves the use of aqueous solutions. For this reason, the researchers used ethanol in their experiments, which allowed him to see the CAHS proteins in action inside cells. CAHS proteins appear to act as cellular scaffolding structures similar to the cell's cytoskeleton. However, they only work when the cells run out of water. In dehydrated cells, CAHS proteins bind together. The cytoskeleton-like structures protect the cell from complete disfigurement due to lack of water, and likely contribute to the remarkable stability of the cells. This process, called anhydrobiosis, can be reversed, allowing tardigrades to start life at the very moment they start to run out of water or other extreme conditions. Scientists have previously suspected that there is a protein-based mechanism that keeps cellular structures intact during drying. However, previous studies have only looked for the genetic components of this ability. In the new work, the researchers focused on proteins. Neat biological tricks like these have allowed these eight-legged animals to reach all corners of our planet. From scorching volcanic vents and the crushing pressure of our ocean depths to tropical forests and icy tundra. Everything about tardigrades is fascinating, says Kunieda. The extreme range of environments in which these creatures can survive leads us to explore never-before-seen mechanisms and structures. For a biologist, this field is a gold mine, emphasizes the scientist.